medicine is the scenario where you custom made medicine for each and every patient. Okay, so to reach that goal of customized medicine for each and every patient is a very long term goal. Today we are not having this kind of facility. Okay, we do have some customization. Let me tell you which kind of therapeutic customization we have. Consider that there is a kid who is suffering from fever. So he will be given or she will be given a dosage of 300 grams of 300 mg of paracetamol thrice a day. Okay, because his body weight is low. Now, in a full grown individual, what will be the dosage? The dosage will be 600 mg per 600 mg at least 3 times a day. Okay, so this is a very nominal customization that has been there. But over the time, we have understood that this much of customization is not enough. So you need to do a much more customization than that. So I give you one more example why this particular personalized medicine was very hot topic there. It is just after the COVID. In COVID, what happened? People saw that there are people without any comorbidity. If there is no comorbidity also, people get succumbed to this thing, COVID, very severely ill. But on the contrary, some people will comorbidity also, they will not get affected severely. Why? Because there are some specific genetic makeup which ensures that they will have preferential removal of the virus from the circulation. Okay, these are all under study. Now, this is for an infective disease. Okay, now there are some lifestyle diseases as well. Okay, understood. What are lifestyle diseases? Lifestyle diseases are hyperglycemia or diabetes mellitus, then hypertension or blood pressure, and multiple other. These are the two main lifestyle diseases that is on much talk today. So it was been observed that there are some minute this thing what you call regulation at the genetic level that are there in terms of microRNA, in terms of epigenetic regulation. What is epigenetic regulation? Is there anybody who can give an idea of epigenetic regulation? Whatever you know in the chat box, that will be great. If there is anybody, what is epigenetic regulation and what is the snippet we are showing you? This is the integration of all technology for the benefit of human being. There is integration of cloud technology, there is integration of 5G and what else? Internet of Things, IoT. That is absolutely perfect. You are correct. DNA methylation. Now, how DNA methylation affects? This is basically affects through the what do you call this regulation of the histone cycles. Okay, so what exactly happens if there is a mechanism to preferentially express a particular kind of DNA by means of methylation of the DNA as well as acetylation and deacetylation of the histone. Okay, as you already know that if there is a DNA which is expressing, it must be in euchromatin situation, in euchromatin form. If it is in heterochromatin form, you cannot able to express that DNA. Now, how this euchromatin heterochromatin transformation takes place? By means of epigenetic regulation. Okay, there are multiple mechanisms for this. Okay, so it has been observed, and then this particular snippet is about integration of Internet of Things, integration of biomedical devices which will observe continuously your heartbeat and all the vitals by means of a wearable device which is similar to your smartwatch that can connect to a third-party network and then transfer your heart rate to a server, and the server can monitor whether your heart rate is normal or not, and eventually can suggest how long you should walk every day. Now, this is also there. So this is the technology of the electronics people and computer science people who will work on that. And what is our cup of tea? Our cup of tea is epigenetic regulation. Madam, please change the slide. Next one. Okay. Now, in order to study epigenetic regulation by means of machine learning, there has been a severe development where there are integration of multiple different algorithms to a single package. Okay, because epigenetic regulation, no, it's the regulatory mechanism as well as the end result is very complex. So this is a very good snippet where customization of this thing where uh, customization of this medical therapeutics is dependent on multiple different algorithms. They are using support vector machine, they are even using artificial neural network, and they are also using some other tools and technologies. So this is a snippet which is based on pattern recognition and all other machine learning algorithms packaged in a single single product. Okay. Now next one, madam. In the same line, like 2020, 2021, we had submitted one patent where we will be exploring the effect of mutation by means of computational simulation. So we have already submitted it, and we are about to get the grant, and eventually and subsequently we are also hoping for make it into a complete product and put it into the commercial use. Okay. So, madam, now I would request you to play the video that we have shared with you in mute condition.
Okay, sir.
Coordinator, madam, in the mute
Yeah.
condition. Yes.
Yeah, okay.
So, in this video, we shall see that what are the wet lab modalities to do high-end biomedical research, and how we can replicate the whole thing in a computer-based platform, that is in simulation, bioinformatics simulation. So we'll see the video first, where we'll just make you to understand that how detailed the work should be if you do the same thing in wet lab, and how you can reduce the cost to a significant extent if you trust the computer science capabilities. Full screen, madam, in mute condition. Okay, so this is a company which works on multiple different aspects, including this molecular diagnostics. So this is a usual lab where people will be working on multiple different aspects of drug discovery, translational medicine, clinical development, precision medicine. That is what we are interested in. Okay. So usual day is how like what they will be studying gene expression by means of multiple different things, including microarray, then a real time this thing, what do you call, PCR and all such things. Okay. So these are different areas where it should have, if you want to build a lab like this, there should be these the different areas in your lab, and you should have these many equipments. It could be anything from your next generation sequencing platform of Illumina or Roche. Then you should have a high throughput ELISA by BioRed or something. Then eventually you should have a microarray analyzer. Then you should have a fluorescent microscope, the usual PCR, RT-PCR, and what else? You should have treatment facility for biopsy sample. Then all sorts of them, protein sequencing, protein crystallization. This is a 190-degree this thing freezer with liquid nitrogen, and all sorts of freezer. These are very expensive. Okay, then there should be a proper genomics lab. Okay, so this is not the single genomics lab. This could be many things. Anatomical this thing where microtom will be there, where biopsy samples will be done. Now this is a biopsy. Biopsy sample will be done. Then you have to isolate DNA from that, and then you should do the sequencing. If there is a mutation, you should do the mutation profiling on multiple different things. Here, the biopsy sample is being handled like this. Throughout that day, there will be so much of task. If you want to do precision medicine, okay, precision medicine means a single patient should have a single type of drug. Okay, it could be a monoclonal antibody, it could be a therapeutic protein like growth factors. Okay, it could be something like this thing hematopoietic what do you call factors. Then it could be stem cells, genetically engineered stem cells, diseases like for skid or for thalassemia or for aplastic anemia, many diseases where there is no other option but to transfer the genetically engineered stem cells. Okay, so this is gross usual high-end biotechnology lab or biomedical lab where you have to have this many equipments and all such things. So they are showing it genomics lab, but it is not genomics, it's integrated to everything. Okay, then after post amplification you should visualize the DNA, fluorescent microscope should be there, and multiple other things. Okay, and then there should be a huge integration with the robotics. Okay, why robotics integration should be there? If you want to handle a lot of sample, then there must be some automated process that is going on. Okay, it is an automated ELISA that is going on there. Okay, so this should be the dimension of the lab, which is very huge, very huge. Okay, they are taking the DNA sample as well as the this thing blood sample from where they'll remove the buffy coat, and then do the isolation of DNA, genomic DNA. Then they are processing and subsequent sequencing, and then multiple other things is going on in the whole. As you can see here, this is the fluorescent tag that is being analyzed. Then there should be FACS, fluorescent activated cell sorter, which will identify how many ploids are there, is there any abnormal DNA there. Then there should be HPLC, which will identify if there are some mutant protein like mutant hemoglobin. As you know, multiple different types of hemoglobins are there: hemoglobin epsilon, hemoglobin F, hemoglobin A, hemoglobin beta. All sorts of things should be there. Okay, thank you, coordinator madam. Now we'll go to the hands-on. Okay, it will keep on going because multiple different things are there. So I am sharing the first tool today. Shagnik, if you have already joined, share the first tool and share your screen as well. Click on the first tool and then share your this thing, what do you call, screen. Let us see what is the tool today. Okay, so this is a hybrid model of DNA binding residue prediction. Okay, so what exactly happened? Who can tell me why DNA binding residue prediction in a protein is important? It is important because I give you a little this thing basics. I'm going to the basics. So what exactly happens during when we talk about gene expression? Now, how exactly gene expression takes place? There should be a gene, there should be a promoter region where the RNA polymerase will come and bind and it will start producing the mRNA. Okay, now there should be some inhibitors, there should be some inducers and multiple different things which will regulate the gene expression. Now, once the mRNA is being formed, it should be processed by means of multiple different things, including splicing and all other things. Post-transcriptional processing is also there. And subsequently, the mRNA will bind with the ribosome. Now, that ribosome could be a free ribosome in the cytoplasm, or it could be also the lysoribosome, which is present on the cytoplasm, endoplasmic reticulum. Now, based on that, what would happen is your protein will be formed. Now, in the DNA sequence, there must be some specific what you call consensus sequence, which will enhance the binding of the RNA polymerase to that particular area. Okay, so similar is the story with the this thing also, DNA polymerase also. Now DNA polymerase, what it does, it starts producing copies of the DNA, but it does not start from anywhere. It starts from a particular site, which is usually an AT rich site. AT rich site means this site is very easy to be broken. Since there are A and T, there are two hydrogen bonds, it is much more easier to be broken than that of the CG. Okay, where epigenetic control is usually done. Now, coming back to this, so in any protein, if there are some DNA binding residue, what does it mean? It essentially means that some or the other way, it will help in epigenetic regulation. Till now, this thing is clear for everyone. Then we'll go ahead. Just write yes or no in the this thing in the chat box if these things are okay. So what we did, we understood that there is a very important this thing of precision medicine, where essentially talks about this regulation of gene expression. Now, regulation of gene expression could be by means of multiple different things. One such thing is binding of proteins on the DNA. For example, you want to replicate a particular kind of DNA. Okay, for that there should be a ORI site. Now that ORI site is rich in ANT. It should not be any AT rich sequence. There should be some consensus which is compatible to the active site of the DNA polymerase. Then only DNA polymerase will start working. Similar is the case for RNA polymerase. Now RNA polymerase will scan the whole DNA. Okay, you close your eyes and think like that. Now RNA polymerase is a multidimensional protein which is scanning your DNA. Now that DNA is already in euchromatin form. That means no histone is there nearby. Therefore, it is very free. Where inside the nucleus. Now the RNA polymerase is also present in the nucleus, and then what it is doing is it is scanning the whole DNA until and unless it reaches a point which is known as promoter sequence. Now that promoter sequence must have some specific residue, specific what we call is three-dimensional motif, which will be bound by the RNA polymerase. Now RNA polymerase is a protein. RNA polymerase is a protein, and RNA polymerase must have some residue, must have some amino acid residue which will interact with the consensus sequence of the promoter region. Understood? Now this is the background study that we already know. Now what we do not know is why it is important to identify DNA binding residues on the surface of a protein. Okay. Now for that, what we will do is we'll identify some of the proteins which we think that we'll able to do what we call DNA binding capabilities. Now, Shagnik, go to the this thing NCBI PD this protein. Go to NCBI protein. Fine. Now here you select protein, all databases. Now here you select protein. Okay. Now here you write. Just hold on. Okay, RNA polymerase beta subunit, like this you write for E. coli. RNA subunit, RNA polymerase E. coli, like this you write. RNA polymerase E. coli. No, only RNA polymerase only. Then click on it and search for the lowest molecular weight protein. Hmm. 152, that is good only. Hyperviridae or hyperviridae. No problem. Let us see. Select. Okay. Now there are some quick controls that a feature for developing the model. Is it hybrid? We are selecting hybrid. Then select the threshold value. We have selected the default threshold value. no need to put this thing, what do you call, email. If you want, you can get it, so that the result will directly transfer to your email ID. Then submit the sequence for prediction. Now what has happened? Your order has been deposited to the uh, a mainframe computer of a particular university. Okay, and then you are getting the this thing, what do you call, analysis done without downloading your the whole software, because you are using computational tool of a third party. Okay, only through by means of the, what do you call, internet. So it will give you the result. It will take a little time to calculate. There are multiple different other applications of epigenetic regulation as well, other than precision medicine. So I give you an example of antibiotic resistance. Okay. So can anybody tell me what exactly happens in antibiotic resistance? Now in antibiotic resistance, basically we are talking about multi-drug resistant bugs or bacteria. Now this thing is, I think I have already spoken. Antibiotic resistance or drug resistance is not only antibiotic resistant. It could be chemotherapy resistant things also. Now there are, please listen also carefully. I do not know whether you are aware or not. I think you will be aware. Please make notes as well. So DNA synthesis is a two-step mechanism. There are two types of DNA synthesis. One is called salvage pathway, another is called de novo pathway. Most of us already know about. Now, de novo pathway is very difficult to perform. It takes a lot of time because it starts from a nitrogen base, a basic nitrogenous molecule like ammonia kind of thing. Then it completely builds a nucleotide from scratch. Now, this kind of DNA synthesis is very difficult. DNA nucleotide synthesis is very difficult. Now, that nucleotide, if it is been formed, it is stored in the nucleotide pool, and whenever DNA replication takes place, no, at that point of time, for example, in PCR, what you do when you are trying to do a PCR, you are adding 5-6 things in your PCR tube. You are adding your this thing primers, forward and reverse primer. You are using Taq polymerase. You are also using MgCl2 as the cofactor of this thing DNA polymerase, because in DNA polymerase active site there is a prosthetic group of Mg. Many a time, since the prosthetic group is not covalently linked, please listen carefully. What I'm saying that in PCR tube you are using Taq polymerase, you are using the forward and reverse primer, you are also using magnesium chloride of a particular strength, you are using a buffer, and you are also using 4 different nucleotides: adenosine, guanosine, cytosine, and thymine. Now, this is in vitro replication. Everyone knows PCR is nothing but replication in vitro. Means you are doing the replication thing only, which is usually done in cell. You are doing it in a test tube. Okay. Now, in order to simulate the whole situation, like the test tube, like the cell, you should give him everything. You should also give a this thing DNA polymerase as well. Now that DNA polymerase should be highly thermostable, because if it is not thermostable, wrong thing may happen. Wrong thing means what? There should be wrong insertion of this thing. What do you call nucleotides? In order to reduce that, you should do it in a high temperature. Now, while doing the high temperature, since DNA polymerase is a protein, it will instantly disintegrate. Okay, although you are putting a Taq polymerase, despite of it, it will get disintegrated, and therefore, to maintain its stability, you are using MgCl. Okay. Now, what would happen if this thing, this what would happen if there is a scenario that you are not having the nucleotides A, T, G, C? Then what would happen? All other things are there in the PCR tube. If you do not add these 4 nucleotides, nothing will happen, isn't it? Why nothing will happen? Because with whom you are doing the replication, with whom you are doing the polymerization, isn't it? You do not have the building block itself. to make the building block inside the cell, there are two pathways. One is salvage pathway, one is de novo pathway. Please make notes. Now, de novo pathway is very difficult to perform, but salvage pathway is easy. Why salvage pathway is easy? Salvage pathway uses all the useless DNA that you have already inside your cell or inside your system. So from where the useless DNA comes? Useless DNA comes due to the repair mechanisms that is every time going on inside your cell, inside your body. So every time some nucleus is getting degraded. Now where the nucleotide goes? That nucleotide gets stored inside a nucleotide pool which is already there inside a cell. Okay. Now to maintain salvage pathway, there is a very important thing that is needed, which is known as folic acid. Please listen carefully. I'm talking about drug resistant cancer or chemotherapy resistant cancer. Please understand. So what happens? There is a very desperate need. Folic acid is only useful for that reason only, to produce DNA. Okay, in order to produce DNA inside the cell, folic acid is needed. Folic acid is one of the 12 vitamins that is there, B complex vitamins. Okay, now a Indian scientist, being I'm an Indian, I know that all of you are not. Some people are there who are foreigners, but please understand this thing very clearly. There was an Indian scientist during 1930s. He predicted that if he could make a folic acid analog, he can treat cancer. 1930s, please remember, when we had no PCR, when we had no microarray, when we had no fluorescent microscope, nothing. Now think the level of what we call genius of the person. Can anybody name the person whom I am referring to? Is there anybody? His name is Professor Yala Pragada Subbarao. Why Subbarao? He was an Indian who studied in this thing, Stanford, and remained throughout the life in there only. Okay, in USA only. He was the discoverer of folic acid analog, which is known as aminopterin. How many of heard of aminopterin? Aminopterin is the first drug which is useful for cancer. So I'm talking about 1930s. Now today, if you give aminopterin to a cancer cell, nothing will happen. Why? Because the cancer cell has adapted to that thing. The folic acid analog is been given and nothing happens. It metabolizes the folic acid analog and eventually makes it folic acid only. And as you know, cancer cells are in desperate need of folic acid because it is in desperate need of to replicate. Uncontrolled replication of this thing cell is called cancer. So during cancer, there is a desperate need of folic acid, and if you give folic acid analog, cancer cells will die. That is a thumb rule, but not anymore. Why? Because most of the cancers has become drug resistant, chemotherapy resistant, and therefore newer drugs, for example, monoclonal antibodies, magic bullet therapy, this and that is needed. Okay, tomorrow we'll learn about breast cancer therapy and all a little bit more in detail. So what I'm trying to figure out is to make you understand that this kind of antibiotic resistant or drug resistant is also something have to do with what you call epigenetic regulation. These days we are finding it that there are minute epigenetic regulation. What is happened to this? I don't understand. Your result is stored for 5 days. You can book. Acha, why this is taking so much time? Shagnik, what you do is you copy this yellow, this blue, this thing. What do you call is copy. Do not click it. You copy this and save somewhere in some place in your email or WhatsApp or something. Because after some time, no, you click on this and see whether the result has been there or not. Because we will be losing out of time if you stick there. We will go to the next of the other this thing. So simple. Net. What is the net? What is the story here? This is a tool which is made up of an algorithm which is called position specific scoring matrix. It is a data science tool which uses computational technology as well as statistics to predict whether a DNA is having protein binding residue or not. Now, why it is important to know whether a DNA sequence, why it is important to know whether a protein is having DNA binding residue or not? Because if there is some protein DNA binding residue, you can be rest assured that some or the other way the protein is useful for epigenetic regulation. I'm repeating one more time why it is necessary to understand if a protein sequence is having DNA binding residue. Why? Because if a protein is having DNA binding residue, it will bind to DNA or either inhibit or what? Opposite of inhibit to that DNA. This DNA expression. Either it will inhibit the DNA expression or the gene expression, or it will enhance the gene expression. Are you getting my point? Like IPTG. Okay, IPTG, what it does, it enhances the production of lac-operon in E. coli. All of you know. So something like that. Are you getting? So if you want to study drug resistance, antibiotic resistance, chemotherapy resistance, and all such things, this particular snippet is very, very, this particular tool would be very, very important. All the tools will be shared to you. Do not worry. Understood? So it takes a lot of time to this thing. I would have given my email ID there so that it could have been a better choice. Acha, Sagnik, what you do now? Sagnik, what you do now? You once again click on the link. Leave this link. We'll do the same thing while I'll put my email ID there. So Sagnik, are you getting what I'm trying to say? You open one more browser and do the same thing that you did already. Now here in this case, you add my email ID there. Just hold on, give me a little time. I have shared my email ID in the chat box. So what I'm trying to say, I tried to say that antibiotic resistance and drug resistance are very important, this thing of study. Okay, now in order to do this kind of study, where you are thinking that there is a epigenetic regulation and I need to figure it out. At that point of time, if you want to do it in silico, that is much easier. In the contrary, if you want to do it in vivo or in vitro, it will be a huge task. Okay, and if you belong to this thing, if you belong to if you belong to a lab which is resource limited, it will become impossible for you to study epigenetic regulations for any kind of drug resistance. Okay, excuse me. If no, okay, that link is not opening. Achha,
No, sir, it is not opening. It is going to some other things.
Okay, no, what you do know? No, I understood what has happened. So what you do know? You go to this thing. You go to that where it is running, taking buffering a lot of time. No, you go to that page. You are there. No, now there must be some home button. No, there is no home button. There is a
I
home
can
button.
go back or I
Do
can
not click it. Do not click it. Left click and go to one more tab. Other tab. Open in a different tab. Here you do. Now here also it is the same. I think now it has started. Now hybrid. Go now. Do the same thing. Paste the same link. Yes. Here you do the same thing and you put my email ID. So if it takes much more time, you need not to worry. We will go to the next. Okay, you can put your email also, no problem. Any email, just
so
let us know.
I'm putting your email on.
Okay, fine. Okay, you put a T. You put a T instead of an Y.
Okay.
Go back. Fine. So I'll request our coordinator, madam, to now click on the link of the research paper. We'll discuss a little. Shagni, you stop
Should I
the
stop
screen now.
sharing?
Yes, you stop sharing, madam. Will go and we'll give them an idea that how exactly we can study this antibiotic resistance by using completely in silico tools and eventually also get published. Coordinator, madam. Yes. Accept all the cookies. Accept all the cookies. Fine. Now this was the study that we made long back again 2000 and very early 15, 14, 13, I think. So here we have explained how drug resistance is working in a particular kind of fungi. Okay, very dangerous fungi. It causes multiple kind of diseases. Okay, when we talk about fungi, it seems that okay, it is not very this thing. It's just a mushroom, nothing to worry. No, there are multiple fungi which are very very destructive. Destructive so much that if you are a immunocompromised patient, if you are a AIDS patient, or if you are going through some chemotherapy where your bone marrow is also compromised, in those cases, this particular fungi that is called oh yes definitely which is called Candida albicans. It is having large scale what do you call this widespread drug resistance. Okay, now the reason for the drug resistance is mm, the reason for this drug resistance is a prey predator kind of relationship. I'll just explain the prey predator relationship and then I'll come back to your question why it is 0.5. Okay, Sheikh Zishan Rahman, this I'll answer just right away. I'll just explain this, otherwise I'll forget. So, how exactly drug resistance takes place? All of you are already aware, but I'll not leave the chance to discuss this in a nice way. So, what exactly happens? There is a prey-predator kind of relationship. Now, how prey-predator kind of relationship takes place, do you know? In ecology, this is a very useful piece of information, which is very important to be understood by by biomedical people, biomedical scientists or researcher or students. So in prey-predator relationship, what happens? You know, the nature will support both the prey and the predator. Consider this: that there a tiger is running and trying to catch his prey, which is a deer. So once upon a time, the deer had very long horns. Okay, now these long horns are very difficult to handle because it increases the weight of the head as well as it gets trapped into the jungle while running. Okay, so what has happened initially? Those this deers who had very large horns, they used to get killed by the tiger, and eventually the nature has understood. Okay, if the horns remains longer, then this particular breed of deer should be present in somewhere in polar regions, not in a tropical forest like Amazon. In tropical forest like Amazon, he or she, the deer, must have smaller horns. Now, when the nature has made smaller horns in Amazon, the speed of the deer has increased substantially. Now, what has happened in the contrary? The tiger was not able to catch the prey, and eventually all the tigers were getting dead. Now, that is also nature cannot this thing what we call sustain, because nature, what does it want? It wants variety, it wants diversity. The in this thing, why the earth is there today? Because the nature supports diversity, it does not support monoculture. Monoculture, human beings are practicing monoculture, which is very horrific thing for the nature overall. Okay, in Indian subcontinent there are a kind breed of this thing, chicken, which is produced in huge scale, and due to the breeding of this particular chicken, uh, developing countries like India and this particular area of the Southeast Asia can able to have animal protein, because animal protein is very costly. But what is the problem there? If a single bird or single chicken get infected by a virus, the whole breed gets cleared, gets eliminated, because they are exact copy of each other. There is no diversity at all. In the contrary, the same chicken of a different genus is thriving in the jungle, which is called local, or called the wild variety of chicken, because that virus cannot do anything to that particular the other breed or the wild type breed. So what does it mean? It means that nature supports diversity. Now here what happens, nature what it does is, it reduces the size of the thorn, at the same time it also increases the speed of the tiger, and his muscles are also increasing, and he also learns so many tricks so that he can somehow catch the prey, and everybody remains. Nature does not support a single breed, it supports everyone. Here also this is the story is the same. So what we are trying to do, we are trying to eliminate this particular organism, but nature does not know that this is harmful for most of the things. But nature does not know, because nature is only supporting everyone. So this bacteria has got an ability to flux the antibiotic that you are giving. In this particular case, it is azole drugs, azole group of drugs, which it has given a ability. This particular organism, Candida albicans, what it does is, it preferentially eliminates by a proton pump. Okay, by using ATP, it is pumping the antibiotic outside so that it does not harm its cellular integrity. Now, this particular paper has explained this. We have not studied epigenetic regulation, but we have studied the other thing. Okay, now we have seen, not we, the fraternity has found out that drug resistance is not only the change of the structure of a particular protein. It is more than that. What is that? This is epigenetic regulation. Some or the other way, a particular protein is getting overexpressed, or some or the other way, a particular protein is not expressed at all. Now, this is called epigenetic regulation. So in order to study that, we tried to do a little hands-on with the first tool. Now we'll go to the second tool. Now coming back to that, now threshold is nothing but how sensitive is your this thing. I'll come back to his question. This is Sheikh Zishan. Sheikh Zishan, you asked that why we have taken 0.5, why you have not taken 0.8. So if we take 0.8, what would happen is, it will only identify, in 80% probability, it will identify only that protein sequence which have many more sequences. There should be a large domain in the protein which binds to the DNA. Otherwise, it will not. Now, if you give 0.5, means 50% probability, what it does is, if a particular protein is having only 2 or 3 residue which can bind with a protein, which can bind with the DNA, it will give you that result. Are you getting? So 0.5, 0.8, the threshold level is nothing but the threshold of sensitivity, or not efficiency, sensitivity. So how sensitive your prediction is? If you give 100%, what would happen? It will only identify the proteins as DNA binding, which is binding the protein. You will lose many data, because epigenetic regulation, no, it's very intricate. It is not that like DNA polymerase or RNA polymerase, which everyone knows that is definitely binds DNA. Zishan, are you getting my point? If you have got it, you write down okay. Fine. So this is what I wanted to convey to you. So why you should not take 100%? Because if you are doing only every time RNA polymerase and DNA polymerase, then it is okay. It will give you correct prediction. But what do you want? You want to see, you have isolated a protein. Now you want to see whether this protein is responsible for some or the other kind of epigenetic regulation. At that point of time, do not put it 80%. You put it 50%. See the result. Then you increase a little bit. Okay. Then you do a wet lab study and some other things, do literature research, and eventually you increase, and there should be some justification. Understood? So that was the story I was wanted to tell you. Now going to the next one, Shagnik, go to the next this thing tool that we already have. Click on the link. Shagnik will present now. Great. Now this is a RNA mining this thing tool. Now here what happens? Just hold on. Okay. Now consider that you have isolated a microRNA. Okay. So microRNA is very much in demand these days. Okay. Why? Because we have found that there are some microRNA which binds with an RNA and makes a dihybrid, and eventually the RNA is destroyed by the internal nucleases. So do you know what are nucleases? Inside the cell itself there are nucleases. Okay, for example, RNase H, there is a, how many of you have heard about RNase H? RNase H, I'm writing it, RNase H. It looks like exactly this in your molecular biology textbook. How many of you have heard about RNase H? RNase H is a protein which preferentially destroys RNA whenever it finds a double stranded RNA. Are you getting? Now consider this. Now consider this. Your body has made one RNA. Now what is the long-term objective? The RNA should find a ribosome and it should get expressed. Now this is the idea. Now what would happen if a complementary RNA is also formed somewhere, and that two RNA comes together and binds with each other due to the Watson-Crick base pairing, and what would happen? Inside the nucleus, that RNA will get destroyed. Now, do you think that is epigenetic regulation? This is definitely epigenetic regulation. Why? Because you are producing an mRNA to produce a protein. Now, due to the presence of the microRNA, your protein is not getting expressed. I do not know whether you have come across this discussion previously, but this is what today's biomedical scientists are working on. The ability of microRNA to bind with a normal RNA and try to see whether it can do the epigenetic regulation or not. Epigenetic regulation means what? Some or the other way control gene expression. That's all. Whatever way you have. Okay. So now this tool will help us to do this, to understand whether a RNA can produce a protein or not. Okay. So there is a tutorial, but where is the this thing? Just hold on. Oh my goodness, it should be this thing. Download it. Run. Shagni, go to run. Just hold on. Okay. Okay. Fine. Now go to NCBI database. Here you select nucleotides, and nucleotides. Fine. Now here you write down uh, 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 most of the things are this thing only. This coding proteins only, from where you can get the non-coding RNA. Achha, anyway, here you write down this thing, insulin. Ah, any insulin. Enter. Okay. Everybody is CDS only. Complete DNA sequence. The mRNA, precursor mRNA, low. First one only, 432 BP. That is good. Faster. 432 BP. Getting know what we are doing? Some other animal. I do not know which animal is this. It's insulin's mRNA we are selecting. Copy. No. You should not copy. You download. No. You have to download it. Acha. What? You go to send to. You go to send to. Can you see the send to? Click on it. Then clipboard or file. Faster. Create file. It has downloaded, right? Rename it as. You copy this thing. Octodon Degas insulin mRNA. This whole thing. This text you select. No, the text. Oh, no, not this one. Acha, anyway, you can do this also. No problem. Enough. Copy, rename that file that has been downloaded. Some file has been downloaded, surely. See, you download, you this thing, rename it. Fine, absolutely fine. Rename, paste. Choro, leave it then. Sequence first, only single file is there, no, in your computer. Then no problem. Go to this thing, RNA mining, and then upload. Select file, upload. Okay, now click on the organism thing. No, organism thing. See whether this particular organism is there or not. What was the name of the organism? Go to that website again. That was a problem. No. What is the name of the organism? Octodon degas. I don't know. I don't think that it will be there. Let us see if Octodon is there or not. Here you write down Octodon. O, anything? No, 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 no. Oncorhinchus. No, this is some kind of bird or something. It is not there, na? Down, down, down. Only this much. Arre, yaar.
Sir, from only one organism.
I understood that. So what should I do now? It will do ulta pulta. You go to that database again and then select Homo sapiens. Insulin, Homo sapiens. See that side, come to this side. See where Homo sapiens are. There you similarly RNA only you select. RNA only you select. Much longer. This will be the problem now. Oh, okay, found it. Yes. Faster. Partial. Let it be partial. No problem. Download. Now there will be a problem. You delete that thing. Every time it will put a sequence file only. You have to rename every time. Now delete the earlier one, or some or the other way. If you can manage, that is fine. Okay, upload. Sequence 2 it will be. Sequence 1, right? Okay, now here Homo sapiens. Fine, run. Let's see what happens now. Now what is the result? Coding. And the classification of probability is this. Okay. So why it is not 1? 1 is the highest probability. 0.6 is intermediate, more than 0.5, but not 8 or something, isn't it? So why? Because it was a partial CDS, and therefore you got the result very fast. So what is the use of this? What is the use of this? The use of this is if you have come across an mRNA by means of sequencing. Now this isolation of RNA is very tricky, because why, I'm just giving a context. It could happen that you are already aware, but still I'm once again. Now, during isolation of RNA, there is a stage of using RNase inhibitors. Okay, because RNAs are very fragile. These are single stranded, and therefore one more step is there where you need to freeze the sample from where you are doing the isolation of RNA. So if you see the RNA isolation process by any company, you will see that these two steps are definitely there. One, freezing, and the second one is addition of RNase inhibitors. Now, interestingly, most of the RNase inhibitors are gold derivatives. Orium chloride is a RNase inhibitor. Okay, so it is a very tricky, a very difficult to do, and very problematic area for eukaryotic cells. Now, if it is a viral RNA, that is okay. These are longer, very large, but this expression level RNAs are much smaller, and it's difficult to handle and very fragile due to the single stranded nature of it. So this will give us an idea that now Shagnik will do one more experiment a little. Go to that site only, that NCBI only. There you open it. Here you write no non-coding mRNA. See what anything comes. How many things comes? Non-coding mRNA. Non-coding mRNA. enter. What is there? No, precursor, precursor, transplant, splice variant B, non-coding transcript. Then there is Homo sapiens, RNA, mRNA for B. Acha. 431 BP I can see. There is also non-coding. Let us see how the tool performs. They are saying it is non-coding. Open it. Now go to the same tool, copy, paste, or download. Download. Now see what happens. Again, click on the run. Same. Now, just for the sake of study, organism, Homo sapiens, and then see what happens. Fine. Good. Arey, what a probability. That's good. Okay. So I'm doubtful. Acha, Shagnik, when you, acha. There is a catch here. The catch is when you download FASTA, no? Acha, Shagnik, can you able to edit the FASTA? So do one thing. You go to the downloaded file, number 2 file, that
Yes,
means
sir.
sequence file, and
Yeah.
open it by text.
Okay.
Are you getting what I'm trying to say? Good. Open it by text and delete that this notion, the rest of the other thing. When you just keep this thing, you open it. Open with, you'll do like open with. Then go for text. Where text is there? Notepad. No, notepad. Just once, yes. Open it. Good. You do know, you delete everything. You keep that greater than sign and you delete all the other things. Okay. Now, once again, do the same thing. Let us see what it does. Save and then, okay. Good. Very good. Close. ठीक है. तो save करो इसको. Save. Fine. Close it. Now, once again, do the same. Let us see what happens. Now it would have already added it into its a training set, no? Let's see what happens.
Sir, it is not running.
Hmm?
It is not running.
No, it will say something. No, why it is not running? Achha, you
So
do one thing.
on the option on run RNA mining, there is a disclaimer like stop.
No problem. You do is you go to the run thing again and open it in another window. Go to run in another window. New tab. Now you do the same again. What is saying? Good. Okay. If it is that smart that it has already added the whole sequence into its this thing training set, this is possible. Otherwise, it is possible because it is really a good tool. Good. Okay. So what I thought is, because in the FASTA itself there are some information that this is non-coding, this is this and that. We have edited it. Despite of it, it can identify. This is stable only. Very good. Okay. So this is another good tool which will help us to identify whether an RNA that you must have sequenced by Illumina or by Roche or by any platform, that you are having this, what do you call, or by quantitative PCR, that you are having a very good tool to identify whether you are having a non-coding RNA or coding RNA. That is very good. Now going to the third one, Shagnik, till now very good. You have done everything that I wanted. Now going to the third tool. What is the third tool? Okay, just read it out. Oh,
The
acha, acha.
auto
Okay, okay.
APL.
Understood, understood. As I have told you earlier, there is a process called post-translational modification. Now, what is post-translational modification? Once the protein has been built, very clearly, please listen, 5 minutes more. Once the protein has been built by endoplasmic reticulum, the job is not yet done. Why the job is not yet done? It is not yet done because there are some trimming and there are some fine tuning that should be done on the protein. Now, what are the fine tunings that should be done? Consider the case of antibody molecules. Do you know what is the nature of antibody molecules? If you know, you already know that all the antibody molecules are nothing but glycoprotein residues. Now, what exactly is glycoprotein? Glycoprotein is a protein where along with the peptide, you are also having carbohydrate residues. Similar is the case for blood group antigens. Okay, you know there are 4 different blood groups: blood group A, blood group B, blood group AB, and blood group O. Other than O, rest of the others are having real antigens on the surface of the RBC. Now, these antigens are also glycoproteins. So, if they lose the glyco part, they are of no use. Nobody can tell whether it is A or B or something. Understood? Means what? For the correct this thing structural integrity as well as function of the protein should be glycosylated, which is done inside the lumen of the endoplasmic reticulum and Golgi body. Glycosylation, which is a very tricky and very this thing, very what do you call, very complex phenomenon. Now, in order to execute that in all your proteins, there should be some hotspots. Now, in those hotspots only, glycosylation will take place. The addition of carbohydrate residues will be taken place. Okay. Now, this tool. So you can ask me, sir, why this is important for epigenetic regulation? As I have already said, please understand very clearly and make notes. If you have produced a protein which is not functional, what would happen to it? That protein will become inclusion bodies and and uh, phagocytic cells will come and destroy the protein because it is of no use. Inclusion bodies means what? Means disintegrated proteins or this thing denatured proteins which are of no use. Now, what is the fate of these denatured proteins? These proteins should be this thing this should be preferentially eradicated from the system by means of two major types of cells, eosinophils and neutrophils. They'll come by means of pseudopodia. They'll do something which is called cell eating or basic mechanism, or phagocytosis. By means of phagocytosis, they'll eat that thing. Now you can tell me, sir, what is the problem? Let them do the scavenging task and clear the this problem. But while doing so, no, they'll also harm your original correct tissue, because this inclusion body should precipitate somewhere. The abnormal protein, if the hemoglobin is abnormal, it should precipitate somewhere. Where it will precipitate? Who can tell me where abnormal proteins in the blood circulation will precipitate? It will precipitate in vascular tissues. So what is vascular tissue? Vascular tissues examples are kidney, heart, lungs, and all other places. Even heart, in heart, there are specific valves where these kind of inclusion bodies will come, and then the neutrophils will come and try to what, eat that area. So it will eat the inclusion bodies, at the same time it will also eat your tissue, and there will be a large scale what you know inflammation. One such example of inflammation is rheumatoid arthritis, where this kind of inclusion bodies will come and precipitate in the bone joints, and the eosinophil and neutrophil will come and clean that thing. While cleaning, it will destroy your cartilage and you will have severe pain. And what is the medicine of this rheumatoid arthritis? You know, this is nothing but immunocompromised molecules, the molecules which will reduce your immune response. Are you getting? So your protein must be folded correctly. Protein folding is a very intricate and very detailed task that should be done at the cellular level at very high precision. If that thing is not there, your proteins are of no use. They will become inclusion proteins and they will be removed by some scavenging cells like phagocytic cells, macrophages, and then neutrophils and eosinophils and all such things. Are you getting? So this will help us to predict whether protein, a particular protein is correctly made, so that post-translational modification will be done on this or not. So we'll run this tool now. Again, download, and then what else? Download. No,
The
don't want to download.
service retrieve download
Go.
citation.
Okay. Go to service. Let us see what happens. You don't want to download. It is there as usual. So we'll do the same. Acha. Now here, just hold on. I'll select a protein. Just hold on. Shagnik, first of all, click on the example first. Let us see what happens. Which kind of protein comes. Okay, now do run. Let's see what is the result. Oh, please wait for a moment. Okay, no problem. Just hold on. Okay, now what is the result? Let us see. Okay, so many proteins were there now. Okay. Oh, acha, acha, acha. Okay, I understood. Acha, now, Shagnik, go to Google. Go to simply Google. Here you write one letter code for amino acids. One letter code for amino acids. One letter code for amino acids. Enter. It has come. Now go to images. Wait. Click it. Okay, K. Which one is K? Go down. K is what? K is lysine. Okay. So I was trying to tell you that there are some residues which are preferentially this thing for addition of glucose carbohydrate residues. Okay. So this particular tool, what it does is it identifies lysine wherever lysine is there. So every time wherever there is a lysine, it is preferentially adding carbohydrate residues. Means what? If in a protein there is no lysine, it indicates that this protein does not require glycosylation. Okay, that means no post-translational modification is needed in that. Okay, are you getting? So this particular tool, what it does, it identifies lysine residues and how many lysines are there. If in a protein number of lysines are very high, it will what it will indicate? It will indicate that there is a high probability that it should go to this thing Golgi apparatus and then it should come back. Otherwise, from endoplasmic reticulum, it will directly go to the other places. So it is not that all the proteins are glycoprotein. No. What is albumin? Is it a glycoprotein? No. If you search albumin, you will get very less number of lysine. Why? Because there is no need for any glycosylation. But if you do the same for insulin, you will get many lysines. Are you getting? The percentage of lysines will be much more. So today I am giving you a task. Please write it down. The coordinator, madam, please write it down because you have to text it to all the participants. So use two different protein sequences from NCBI protein. The sequence number 1 is insulin. The sequence number 2 is albumin, simple albumin. Okay. Now it could be bovine serum albumin also. It could be human serum albumin also. Now calculate the percentage of Ka which is present in both the proteins by using this particular tool. The link of the tool will be shared to you by me just after the the today's class is complete. Okay, there will be some more this thing also, questions also. So this question today regarding this is, I want to know the percentage of lysine present in insulin in contrast to presence of lysine present in the other protein, which is albumin. So we already know that albumin does not go for glycosylation because there is no need of post translational modification at all. But insulin is highly glycosylated, and therefore it must go to Golgi apparatus for post translational modification. Are you getting why it is important? If you have a protein but you have omitted the lysine, what would happen? Your protein will get destroyed. So you can ask me, so how it could be? It could be because if there is a missense mutation. If there is a mutation and instead of K, some other amino acids come, so how the effect will be there? The effect will be, since glycosylation was not there, therefore the protein has been degraded. And therefore this particular function was not present. Are you getting? So this should be the task, and some more task I'll be giving. Try to figure out the task and try to complete the task in time. If you have any query, please please let me know. Else we will this thing close the day. If you have any query, for tomorrow we will download one software. For that I will give you the link by today night itself. Tomorrow means day after tomorrow on Monday, because we will not take classes on Sunday. On Monday in India it is Sunday now or tomorrow. So what we'll do is we'll come back at Monday, and for Monday I'll give you multiple tasks that you should do, including downloading a software and loading it into your computer. Very simple tool that you have to download, which is a 25 MB size of software, but huge application for that, mostly for to identify effect of mutation. Very interesting study. So that we will do on Monday, and subsequently on the Tuesday we will do all regarding vaccines and antibodies and all tools, all simulations will be given for vaccine design and antibody design and this kind of thing. Okay, if there is any doubt you can this thing else we can call it a day. Coordinator madam, are we good to go?
Yes, sir.
Okay.
Thank you.
Excuse me,
Oh,
sir.
yeah, please.
Sir, I actually have a doubt.
Please go ahead.
Sir, actually, I am a second year undergraduate biotechnology student, sir. So
Great.
it's very hard for me to keep up with the classes and the daily assignments that you have been assigning us with. I couldn't
Hmm.
really complete, or I couldn't even do the second assignment that you allotted us with, sir.
Okay. No, there is no problem in that. What you'll do is you take the recordings, ask the coordinator. She must have the recordings. You take your time, take a little pace, and then you do, because today or tomorrow, no, you have to come into this kind of challenges. Okay.
Yes.
Acha, is there any specific this thing that you want to know, on any specific thing that you are very much confused about? Uh, that you can we
Not
can discuss
really,
now.
sir. Exactly. Like, I'm not completely blank about all the topics. Like, I could
Okay.
understand a few points here and there, but I couldn't keep up with all the points that you explained, sir.
Okay. Okay. For that, no, try to see the video again, and then try to write something. That's our. This is one problem, two problem like this, and then send the thing to us. So on Monday we can able to handle those.
Oh,
And that is very good that you have come across. I wouldn't. Otherwise, I would have this thing. I could not. I did not think that this is a case. Uh,
yes.
is there anybody like anybody else like this where discussions are a little this thing?
Just to
I
clarify,
request you. I
everything we are
request
not
you to
supposed to
give some this thing. Get some in written. If you do something, no, then if you can send it, then I can also go through and I can also understand that where exactly I need to put more focus. But I can
Okay.
tell you that in coming days, if you are sticking to this particular field, if you diversify, that is different. If you are sticking
Yes.
to this field, no, you need to have you will come across this kind of discussion earlier or later. That is for sure.
Yes,
May
sir.
not be the scale, may not be so fast. The pace would be a little. For example, the whole course of BSc will teach you all these things that you have
Yes.
learned these 5 days. whole
Oh,
course of your BSc. This could also be a possibility, but you'll definitely come back to this kind of thing. So you keep these recordings with you and and try to figure out what exactly is going on. You write it
okay.
down that these are my problems from 1 2 3 mane, in a nutshell. By seeing the keyword only, I'll understand what exactly I need to do.
Okay,
Okay.
sir.
Okay,
And
I
also,
think I can help you.
yes, sir. And also, sir, that the new topics that will be starting from Monday, they won't be completely related to the topics that you taught up until now, no, sir.
No, it will be related. No, I tell you how it is related. The theme of this workshop, no, it is the application of computer science for biomedical research. This is the inherent undercurrent of the whole thing. So there we also need to learn a little computer science. Means what are the names of different machine learning, how exactly they work. Is there a possibility that we can also design something by collaborating with the computer science people? Because computer science people are writing codes every day. So if we also start writing codes, it will be all in vain. Why? Because a specialist is already there. Now, in order to make a very good package or software, no, the main thing is you should have very sound background of biomedical concepts, and then you should also think that which is possible to make into a tool, which is not possible. So, in order to give this kind of idea, this particular workshop was being designed. So
Okay.
on Monday we will be teaching about the vaccines and about the antibodies. This is very much a part of biomedical research, and we will use all computer science tools only to devise a vaccine candidate, or to devise or to produce a more antibody. So this will be very relevant only, and this is related to this whole exercise. Okay,
Oh,
this whole 5 days course is about all this. Okay,
okay. Okay,
and
sir.
therefore it will be relevant. I'll give you context. I understood your point now. I'll
Yeah.
give you a slow context, and then we'll enter into the tools.
Thank you
Okay.
so much, sir. And also, sir, the assignments, like the daily
No
task for.
assignments. No, you have to try to figure it out, and you ask the coordinator for the video, and by repeated video over this thing, you give a little, devote a little more time. I'm sure that you will able to figure out. If it is wrong, also no problem. You send it to me. I'll check it out, and if it is substantially wrong, something on a gross misunderstanding is there, then I'll correct
Okay.
it right away. Do not worry. If it is
Okay.
a little bit here and there, I'll avoid it. It's not possible for me, out of the scope. Now, if it is inside the scope, if there is some gross abnormality, I'll definitely make them correct. Don't worry.
Okay, sir. Like I was worried about it won't affect me getting the workshop certificate or not, sir.
No. That is a different story altogether. That the
Okay.
coordinator will tell you. I do not think that it will make much difference. It is only about the value that you are getting.
Yes, sir.
And the certification is, I understand that it's very cheaply priced. 500 rupees is just for a certificate. That is correct only. But my idea of that was not this. My idea was that somebody will get motivated and do further course of action, something
Yes.
like that. I am also looking for a fellow like who are undergrads or just graduate or things like that. But there is a
Oh,
different story. The certification, I do not think that it will have much effect on it. But try to this complete that what do you call
yes.
this exercise and all. It will help you definitely. I'm
Yes, sir.
sure about
Sure,
it.
sir.
Okay. By saying so, I'll conclude this session. Call it a day.
Thank you so much, sir.